Hi, and welcome back. This is Dr. Powell bringing you Anatomy and Physiology on Demand. This is Cardiac and Smooth Muscle, week number 11, Monday class. We have finished cardiac muscle, and so this is the third and hopefully final video. And this one is all about smooth muscle. So let's go ahead and get started. Let's start with what you already know. We learned about smooth muscle cells previously. We know they're fairly small. We know that they're skinny, they're uninucleated, they are spindle cell shaped, they don't have striations, okay? And as opposed to cardiac muscle and skeletal muscle, both of which are forming organs, smooth muscle by itself doesn't form an organ, although in some organs, such as the uterus, it seems like the entire organ is smooth muscle. In reality, it's not. Okay? Smooth muscle can be present just as a single or a few cells acting together in one little place, or much more commonly, they are found in layers, often perpendicular layers to each other. And the perpendicular layers are the types that you would find in walls of hollow organs, where, for instance, you would have an inner circular layer and an outer longitudinal layer. Another place where you find smooth muscle, and which technically is a hollow organ, are blood vessels. But here, all of the smooth muscle would be circular in arrangement, so that when they contract, the diameter of the blood vessel would decrease. Compared to cardiac and skeletal muscle, this is a slower type of contraction, thereby making it being more fatigue resistant and requiring a lot less energy. So when we look at the myofilaments in smooth muscle, just like in the other two types, we still have the thick myosin filaments and the thin actin filaments. However, they are different. The myosin fibers are longer than what we're used to seeing, and the actin fibers do not have any tropomyosin or troponin attached to them. And now we have a third type of myofilament called an intermediate filament. As opposed to actin and myosin, the intermediate filament has nothing to do with the actual muscle cell contracting. It's going to form this cytoskeleton element, which in a few slides we'll see the importance of having this intermediate filament to support the shape of the cell. And something else that's totally different is, I don't know if you've noticed, there aren't any Z lines here, and these do not look like typical sarcomeres, and that's because they are not. Instead, they have something called a dense body, which is just like this blob of protein, the same protein that we find in the Z lines, but it's not in a linear arrangement. And to the Z line, actin will attach to anchor it. And in addition to that, the intermediate filaments, those non-contractile filaments, will also attach it. And so that's kind of how those intermediate filaments not involved with contraction are actually related to the contractile elements of the sarcomere. So here's another picture showing you some microanatomy of 3 adjacent smooth muscle cells, where you can see like the fishnet stocking intermediate filaments attached to the yellow blobs, which are those dense bodies. And then the more gold and dark green, that's going to be where the actin and myosin is. Just like cardiac muscle, smooth muscle only has endomysium. There's no perimysium, no epimysium. But totally different from both of those, there's very little sarcoplasmic reticulum. It's a very simplified amount of endoplasmic reticulum containing calcium, and there are no T tubules. So the mechanism of contraction is totally different once again. So yes, we have actin and myosin, but no, we don't have striations because they're not arranged in sarcomeres. We don't have any Z disc. It's totally different. We have the dense body, and we also have gap junctions. And so if you look at the dense body on that lower right picture, what you will notice is those dense bodies, are actually transmitting contractile forces between two adjacent smooth muscle cells. Okay. And if we have gap junctions, because some smooth muscle does and some smooth muscle doesn't, as we'll talk about shortly, then we would be able to transfer ions from one to the other. And lastly, looking at the bottom of the upper picture, that lowermost cell of the three, you'll notice that there's something there called a calveoli. And so this is a new structure that's only found in smooth muscle. So looking at the calveoli, which by the way just means little caves, it is basically a tiny little invagination of the sarcolemma, which kind of has a clover fieldy kind of shape, clover leaf type of shape. And what is important about it is that calcium hangs out in here. And so it's got tons of little calcium channels right in this area. So if calcium's hanging out right next to all these calcium channels, what that means is now we have an abundance of calcium which can quickly diffuse into the smooth muscle cell, and realizing and remembering that it's much smaller cell, this is how we get rid of having a complex sarcoplasmic reticulum for calcium. So looking at the smooth muscle contraction, when it contracts, it's going to, because it's hollow organs, basically, it's pushing either fluids or solid along a pathway. And not all smooth muscle is innervated. We're going to have two types, which I'll get to very shortly. So some is innervated, and it's an autonomic motor innervation. And then other ones are hormonal, like think your uterus. That's all a hormonal impetus for contraction. So these upper pictures are showing what the smooth muscle cell looks like when it is relaxed. And so you can see those intermediate filaments on the outside giving it some kind of shape. And then when the actin and myosin contract against it, basically the cell is going to shorten. But since the actin's attached to both the dense body and the intermediate filament kind of keeps the cell all intact as one. So it can't just turn into this little tiny blob. All right. So as alluded to twice now, we have a couple of different types of smooth muscle. And of course, anatomy being anatomy, some of these have to have more than one name. And so the first type is either called single unit or unitary or visceral smooth muscle, and the other one's going to be called multi-unit. So let's start with the visceral or single unit muscle tissue. So as the name implies, it's a single unit. In other words, everything's happening at the same time. And as the other name implies, it's found in viscera. Of the two types, this by far is the more common. It's in all of those hollow organs and blood vessels, excluding, of course, the heart, because that's cardiac muscle. Here, what you can see in the picture, we have sheets of these smooth muscle cells. They're all right next to each other. You can clearly see gap junctions connecting all the cells. So if you have an influx of sodium into one of those cells, you can see it's going to be passed automatically to the next cell, to the next cell, to the next cell. And if you notice, there is a nerve going across one side of that. And basically, we have these little areas, the little rounded areas called varicosities. And what happens is when the nerve impulse reaches that varicosity, it is not just stimulating one cell, it can stimulate several cells at once at that varicosity, causing the depolarization of that cell, which through the gap junction will depolarize the next to the next to the next. So they will all essentially be doing the same thing. So that's how the single unit or the so-called visceral smooth muscle tissue contracts. In contrast, Oh, and also want to tell you, this would be autonomic motor because it's involuntary. And this type, the visceral, will respond to hormonal control. So remember, the uterus, a viscera, hormones, it all makes sense. So now in contrast to that, we have multi-unit smooth muscle tissue. And this is found in some very discrete areas, which you know already, such as the erector pili muscles. It's also found in our larger arteries, think your aorta, your pulmonary artery, our larger airways, think your trachea, the first couple of branches off of there, and in the muscles in your iris. So the iris is the muscles that surround your pupil, the dark spot in your eye, and it makes the pupil enlarge and get smaller. And so all of these places, we do not have these cells with all those gap junctions and just one nerve going across the edge. This one, in contrast, has a very rich nerve supply, and the synapses here are akin to those we learned about in the neuromuscular junction, where that varicosity synapses with one individual myocyte. So we have that motor unit complex that we talked about before. Now the muscle in this case actually is not going to generate an action potential. That's different than skeletal muscle. Okay. But it's going to contract in response to the electrical changes happening in the sarcolemma. So just like the other type, involuntary autonomic motor, as well as responding to hormonal controls. So what kind of stimuli can change smooth muscle activity? Well, spontaneous electrical activity can happen in the sarcolemma. Or just like skeletal muscle, we could have neurotransmitters. Or just like cardiac muscle, we could have neurotransmitters released by the autonomic neurons. Or we could have hormones, or changes in pH, or change in oxygen content, or certain ion concentrations, so the osmolarity, osmolality. There are a whole bunch of things, even changes in temperature, plus some smooth muscle has autorhythmicity. Okay? So it's much more complicated than skeletal muscle, and it's much more complicated than we are going to learn in this class. So just kind of stick with the single unit versus multi-unit of concept. Now, before we talk about smooth muscle contraction, there is one thing unique to it I do need to talk about, and that is a substance called calmodulin, which basically is a shortened version of calcium modulated protein. And what this is this is just another messenger protein that binds calcium. Okay? And by binding to calmodulin, then we have a sequence of events happening. So in this respect, calcium, once again, is acting like a second messenger, because it's going to activate the calmodulin. So activated calmodulin then acts as the impetus to change an inactive myosin kinase to an active myosin kinase. And as we learned at the very beginning of the course, kinases phosphorylate. And so what's going to happen is ATP is going to get broken down to ADP, releasing a phosphate, and the activated myosin kinase is going to take that phosphate, and then it's going to bind it to myosin. And so now we have a phosphorylated myosin, which means it can bind with actin. So take a minute to review this part to make sure you understand it, because now we're going to move on to smooth muscle contraction and follow it with smooth muscle relaxation. So I have similar kind of pictures to the ones I showed for cardiac muscle with the numbers, and I left the words here for you. So after the smooth muscle fiber is stimulated, and I have, whoops, my apologies. We have a sequence of events that's going to happen. So the first thing you have to realize at the top, that the calcium for the smooth muscle contraction is coming from outside the cell, from extracellular fluid. Okay, so When it enters, then the cytosolic calcium levels are going to go up. The increase in cytosolic calcium is going to cause a release of calcium that was stored in the sarcoplasmic reticulum. This calcium, not the one that came from the outside, but the one that came from the sarcoplasmic reticulum, then binds to calmodulin, acting as a second messenger, and then the activated calmodulin will activate myosin. Okay. The myosin kinase, it will activate the myosin kinase, which then will phosphorylate the myosin. Okay. And then once the myosin has been phosphorylated, it can form a cross bridge with the actin. So as you can see, it's a little bit different. The ATP is not binding directly to the myosin head. Instead, it's being broken down in the cytoplasm, and the phosphate from that, courtesy of the myosin kinase, is going to be phosphorylating the myosin head. So, a couple other points to remember. The calcium for the contraction is coming from outside the cell, but causing a release of sequestrated calcium, which then puts this series of events into play. Because we have a second messenger system being involved again, the smooth muscle is going to be much slower to contract than skeletal muscle, and it's going to be even slower than cardiac muscle because myosin ATPase is a very slow enzyme. All right, so that's it for smooth muscle contraction, and now we're ready for smooth muscle relaxation. And so what happens here is it breaks back down. Calcium that's in the cytoplasm is going to be pumped back into the sarcoplasmic reticulum, as we have talked about previously, or it's going to be kicked outside of the cell. Both of those are going to require ATP. Calcium that was attached to calmodulin to activate it, that gets broken off. It suffers the fate I just discussed. The myosin phosphatase removes the phosphate from the myosin so that we can't have the activity, and so we will lose our cross bridging and the muscle tension decreases. Now, two important points. The first one has to do with calcium. Part of the reason why smooth muscle is slow to relax has to do with the ability to decrease the cytosolic calcium levels is a slow process. Those pumps requiring ATP to remove calcium from the cytosol, they're slow acting, okay? So we'll take some ATP, send calcium out. We'll also send calcium out, bring some sodium in. We'll also send some calcium back into the sarcoplasmic reticulum, but it's not a fast process. And the second thing has to do with the myosin and its cross bridge. Even though the tension decreases, it does not immediately separate, as what happens in skeletal muscles. As soon as the ATP binds, then the cross bridge is broken. Here, instead of that type of mechanism, we have something called a latch bridge mechanism. And so this is what enables myosin to stay attached without requiring any more ATP. And so basically, I've kind of broken it down into three little parts on the pictures and put some words there so that you can read them on your own. The details of this are not super important. The gist of it is the tension is maintained and can be maintained for prolonged periods because this disassociation, this breaking of the cross bridge happens very slowly. Therefore, the cycle of contraction to relaxation, the contraction's a slow contraction and the relaxation is even slower. So this explains why contractions in labor can last so long. Right before birth, the contractions are lasting about 90 seconds in length, which is an incredible period of time. All right, so I've included this slide here so that you can pause the video if you want to, and you can talk. On the left is smooth muscle contraction, and on your right is smooth muscle relaxation. So go through it and make sure you understand it. And the last concept relating to smooth muscle has to do with what happens when you stretch it. Just the fact that you are stretching smooth muscle is enough to open those calcium channels because they're mechanically gated, they're not voltage gated. The second concept is a concept called plasticity, which means that the smooth muscle can adjust the amount of tension depending on the stretch. In other words, it may have a resting length, but if there's tension put constantly at that resting length, it may increase its resting length to decrease the tension. And the third concept is, as opposed to other muscles, which definitely have a much narrower, that optimal range for contraction that we saw and we spent so much time learning about in skeletal muscle, here, smooth muscle contracts very forcefully if it's stretched or if it's shortened. So in other words, it's not just at the optimal length, but you can go 50% above or below the optimal length and still get a very forceful contraction. So with that, I'm done with smooth muscle. I have words here so that you can compare it directly with skeletal muscle, which you know so much about. And at this point, I want to thank you guys for continuing to work so hard. I realize it's very difficult for everybody. And I will be sending out links for when you can contact me by Zoom as a, a regularly set period. It's probably going to be pretty much every Monday and Wednesday, right around when office hours would have been, just for some continuity. So if you have a chance to watch these videos before you come, that would be fantastic. Look forward to a presentation on the nervous system next, because with this, we are done with the muscular system. Thank you.